So, good evening. I welcome you all in this science dialogue series. As you all know, this initiative is for dissemination of knowledge in bioscience stream. And in this series, as you all know, today's speaker is very renowned. He has served at Biochemistry department, MS University, Baroda. You all know that university, that department is run by father of Nobel laureate Venkat Ramakrishnan. His father started that department. So this department is very well known. So sir holds there as a HOD and teacher also, assistant professor also. After completing his PhD from TIFR, Tata Institute of Fundamental Research, sir has also served as a director in science stream of Nirma University. Recently, sir retired and sir served in different private university also after retirement. So, those who are from Gujarat, no need of his introduction. Those who are from outside Gujarat may not have idea about sir. Sir has served as a teacher more than 40 years, and his PhD students are serving as a postdoc fellow faculty in all over world, not only in India. You have also attended lectures from his student. I have also spent 2 years as assistant professor under his leadership at MS University Baroda. I have used his lab also for my fast track young scientist project completion and execution. With this brief introduction, I welcome sir, Professor Naresh Kumar sir, in this science dialogue series. We are grateful. Sir, are you here? It's fine, sir. Yes. Welcome, sir, and we are grateful for your contribution and for accepting my invitation, sir.
Yeah. Shall I start?
Yes, sir. Yes, sir. I have
Okay.
introduced to the participants. आप निकल गए थे, sir.
Okay. Okay. So,
Hi,
good
this is
evening, everyone. I'm very much pleased to talk about something very interesting for me. That's the metabolism. And Gopalji is giving an opportunity for me to talk about whatever I'm thinking nowadays. So, this is my second topic. Uh, Interestingly, most of these things I have developed after my retirement. So it's a nice thing and a forum for me to put forward my views. Okay, this is I'm thinking about the human metabolism, how it is emerging in this new context of biology, and what I feel is that it is going to be bridging the gap uh, with the physiological anatomical context, whatever is going on. So let me start from the beginning at the what is the current aspects of human metabolism, and all of us may be familiar that the metabolism started with the basic metabolic pathway network and its regulation, and this then we got little bit more insight of this metabolism with respect to subcellular compartmentation, how they happen in the different organs of the cell, and how these different organelle pathways are coordinated with each other. Then the subcellular to cellular level, that giving the holistic picture of the metabolism. This has really progressed over the last 30 years from the basic biochemical perspective to the cellular perspective. And this is with respect to simple metabolism. And additionally, what we have come to know is regulation of metabolic pathways has become at different levels. One is at the what we understood as the signal transduction, transcription level, post-transcriptional, and post-translational level. That is, we're talking about second messengers, transcription factors, miRNAs, and long non-coding RNAs, exosomes. So many things have come into the picture, and that's what they emerged out about last 20 years or so. Then, from the cellular level, we started understanding at the um, organ level specific metabolic pathways. Uh, what is the distinct functions of each organ, and that is how it coordinates the entire physiological perspective of metabolism. That we have seen in the terms of uh, basically concentrating on liver, muscle, and adipose tissue. Liver being the central thing which is coordinating all the metabolic pathways in the carbohydrate, amino acid, so central coordinating component. Muscle is a major organ. So 30, 40% of your metabolism is catered for muscle for all the action, whatever is required. And what we have come to know, realize is when the muscle metabolism, even at the body at rest, is important. And then we have also figured out the role of adipose tissue in this. Adipose tissue, not just as a storage organ. Initially, people felt that it is just a storage organ for the fats, and whenever it is required, it is going to use. But it turns out that it is coordinating multiple organ coordination, very important, in addition to just storage of nutrients, whatever is there. And for all these things, when we talk about the cellular level metabolism, we say whatever goes on inside ultimately is regulated at the transport level. The how much comes in, how much goes out, that is the central coordination. Similarly, when you look at the organism, it is the how much things are taken inside and how much is expenditure in whatever terms. That seems to be the overall point of view. And in that, intestine comes as a central point. Intestines, and I have not given here kidney. Kidney is another component which is coordinates that. So the muscle, liver, muscle, adipose tissue form the central metabolism. Intestines and kidneys function as the organs which are coordinating the inputs and outputs. And the functional wise, of course, with the heart and brain are the major components which use that. But if you look at energetically, the brain is the thing which uses a nervous system almost 20 to 25% of energy and oxygen which is required all the time. And that's a very fascinating point of view. The neuronal metabolism, how much energy is consumed by the brain. And it turns out in the sleep time, where the body, the brain function is much more active, and where nowadays we know the sleep metabolism, how much is important for the every day it is required for the restoration of all the body functions. All the organs have to be coordinated with the sleep level, resting time metabolism. That is also not in terms of only time point, it with respect to circadian cycle, that is the day and night, that coordination is very, very important. So this all we have understood, and we also know a little bit more insight into this, how these things are mediated by growth hormones, growth factors on all those functions. Another, many of before looking at the endocrine systems and everything, basic components of what we talked about, metabolic pathway, cellular level, transcription level, all those things what we given before, this knowledge has come from various systems that I'll give you later. So in addition to this, the very interesting aspect has come in the mammalian systems and animals, which are not the lower system. That is the inflammation and metabolic dysfunction. Inflammation as a means of tissue damage, infections, everything is known for almost 100 years. But the what we call now chronic inflammation, which is very, very low level, that is about 100, uh, uh, 100 times less than the inflammation, what we know of acute inflammation. That is a thing which is associated with the organs dysfunction. And this dysfunction, very interestingly, the what we also come to know is normally three levels of inflammation is there. Basic level of inflammation inside the organs is to ensure that the some cells which are in a bad shape, to repair system to activate. We know about the autophagy, that the low level of inflammation makes that cells undergo autophagy and repair and go so on and so forth. But it goes beyond that, then there is a apoptosis. The cell dies. But in an organ, we know that organ is a very coordinated fashion. You have extracellular matrix, everything, intracellular coordination, everything is important. You cannot have dead cells in a tissue. So in order to remove the dead cells, whatever they be, there is the again immune system comes and tries to eliminate that. So the inflammation is seem to be very important in maintaining the homeostasis. But in some reasons, we know, like what we talked about, the acute infections or tissue damage or something, you have an acute inflammation. But here also, inflammation, you may have some sort of a dietary factors or hormonal dysfunctions, and many things are there that can increase the inflammation in the tissues. And that causes the metabolic dysfunction. And in my last talk, we have talked about the interrelation between the inflammation and metabolism, how it leads to metabolic syndrome. And that picture has come into a part of the current metabolism. Interestingly, the energy metabolism, the inflammation, tissue damage, the metabolic components which are formed over the how this inflammation is triggered on, it is known in terms of damaged tissue components. Simple ATP present outside the cell is a problem. Adenosine outside the cell is a problem. This type of RNA, mRNA coming out of the cell, and mitochondrial RNA coming into the cytoplasm, that many types of dysfunctions are possible. This low level of thing which can be involved in the homeostasis of the cell, and this is connected with the simple metabolic electron transport. What we know is that electron transport is a thing which, at the complex one level, ROS gets generated, and the ROS is coupled with inflammation and so on and so forth. That type of concepts have come, that the metabolism is uh, some integrated with cellular process and some sort of immune function. That is the next level of understanding we have got. And last 20 years or so, we realized the importance of gut microbiome. This is a completely added another dimension to our understanding of metabolism, where the nature of microbes which are present in our body, those are large numbers are there. Numbers are almost equivalent to our body number of cells, and the depends on whatever is in good health. The nature of the microbiome decides our health, and this has been clearly demonstrated by transferring of fecal microbiota from animals to animals, humans to animals, and all kinds of things. They clearly showed the microbes are going to play are playing major role in the metabolic regulation, as well as metabolic dysfunction. These are all the pictures we have understood so far. So of course, there are a lot of gaps in the present in that will go on for a long time. But in addition to that, what is the uh, uh, step ahead? That is what I the my current talk is going to be. And whatever knowledge you have gained so far, these have been used by various experimental systems. One is the in vitro studies, biochemical and enzyme process and organ, whatever things are done. Then it is the cellular level. For cellular levels, we use stem cells, primary cell lines, and organelles. Uh, the studies have been done, and then we have understood this thing. Then we have also used organs, and recently we have also trying to know that, because we know the cell in vitro cells are different from the tissue, what the things are there. So we try to mimic that tissue organ function by creating something called organoids. That is what is going on. In addition to that, we have used extensively dependent upon mouse and rat models, and then of the human studies in terms of collecting samples, or the surgery, what of the tissues have come about. So all those things have helped in understanding the knowledge, whatever we have in both healthy individuals as well as their disease status. This is what we have got so far. But what are the problems with these current approaches? One is the whatever parameters we have used, the biochemical studies or something, whatever we take, is we are coming at a one particular time point and few samples. If you imagine each cell, there are about 10,000 reactions are going on, and your in organ, so many reactions are going on, giving rise to the over 24 hours and so on and so forth. So this is a whatever we're looking at the holistic point of view, this becomes a very limited. Of course, we have to start with what we can do, but that is the one limitation. Second thing is, most of the time, whatever we collect samples are invasive.
सर वो
So,
लाइन को हाइड कर दीजिए, वो नीचे जो लाइन है ना, जस्ट हाइड,
okay.
क्लिक ऑन
Uh,
हाइड.
okay.
यस सर,
Okay.
थैंक यू.
So the most samples, whatever can invasive. So that becomes another limitation. How many times you can collect sample from an individual or animal or so on. That is another problem. Second, most of the information we cannot use humans. So all this majority of the information is come from the animal studies, and animals are and in that animals also we predominantly use males, because we want to eliminate the hormonal variations which are present in the on the physiology. So we use males as a model system. So these are currently that is the we are using, because this is the possibility. We other things become more complicated in analyzing the data and so on. Then whatever the experiments we do for small time period, maybe days to a few months. Where we cannot go for years and years of long-term studies. But in reality, if you look over any simple disease, any disease functions, what we are addressing now, they are taken 10 years, 20 years, 30 years. Uh, so there is a very, very long time. So it is very difficult to mimic. That's the limitation. Second thing is, the whatever metabolic studies we have done, they seem to be uh, systemic thing. We take blood samples. So blood sample is total the whole body as a unit. But when we look at the physiological anatomical context, let's say we'll talk about blood pressure or diabetes, high glucose trigger. So what are the things relating? These are the uh, going to be outcome of specific tissue, specific region problem. But metabolic, whatever is present in the serum samples and blood samples, this is going to be overall perspective. And one more thing which is emerging now, I'm going to stress on that later. There is a parallel circulatory system, which is a lymph that goes from the tissues to the thing. So we don't collect that. That is a very difficult thing. So that is another problem of relating metabolic studies with the physiological anatomical context. See, for us, the skeletal muscle is same, but all skeletal muscles are not equivalent. So that is a limitation of that. Then, in addition to this, what we have realized is there is in any organ, there is be metabolic heterogeneity of cells within an organ. Even in the liver, if you look at the location of the liver, the same hepatocytes in different locations, they seem to have different distribution of functions. And we know that, look at the T helper cells started with that initially. T cells, there are large variation in that. Now we also know the macrophages, the M1, M2, they become very, very important in tissue homeostasis. Now recently people are talking about neutrophils. So there is a lot of heterogeneity is there in the tissues that we cannot mimic that. And this heterogeneity, why it is coming, again, we have some level of understanding where the inflammation and how much oxygen is available, how much glucose is available, how much nutrients are available. That all makes and of course, functional importance of that. So there is essentially what we are saying is there is a metabolic heterogeneity within an organ. We are using one single cell as a cell line or something. Those studies become very, very limited. I'm not saying they're wrong, but that's a limitation. Second thing is as I already told you, that mimicking metabolic dysfunction is going to be very difficult because there's a very long period. So, uh, and if you are using this rat and mice, their anatomy and physiology is different. So, extrapolating, that is the second point, anatomical physiology of the animal systems with the humans. So, sometimes these extrapolations don't seem to be valid. That is why if you look at the top rated journals, Nature and everything, in addition to cellular studies, animal studies, they want human studies data. So whatever we are saying is true with humans. So this is another problem. Um, the In addition to our whatever basic sciences we are doing, we do have a lot of knowledge of human physiology from the pharmacists and doctors. They have a lot of clinical knowledge is available, uh, and that there is a gap in the metabolism to integrate with the knowledge of what is available with the clinician. This is a major challenge for us, and we ultimately we need to fill the gap project from the metabolism with the clinical perspective so that it is useful for the doctors, and doctors, when they understand this metabolism and we integrate, probably our understanding of physiology becomes much better. The major aspect of I'm going to address a little bit later on that again stress on this point. um, the last point, what is many times we try to understand the clinical pathology and population basic physiology of human with respect to population studies. So, but there is a lot of variations in the patient clinical studies and individuals. So if you look at the lot of clinical studies, you see that a lot of clinical studies are published, and there is one meta review comes compiling of all these papers and saying that. If you look at meta paper, there is no clear picture emerges because there's so much of variation is there in the people that is natural and expected because all of us are different. So there's so many environmental factors, so many things factor, that is a real. This is our challenge. So these are the challenges what we have in the current approaches. So so uh, so what is the way out? For this, we have something new things which are coming out. What is that? The what we said the in terms of uh, monitoring part of data and everything, non-invasive continuous monitoring of uh, physiological and biochemical parameters, that is emerging. Some of the simple things are coming as using smart devices. Now we know the continuous glucose monitoring devices are well known, and now people are also thinking in terms of insulin monitoring and various hormonal monitoring and things. So probably these are the things which are going to become very, very important for in the collection of data, and this is going to be completely non-invasive. So that's the trend which is emerging. Second thing, what is going on is that the development of organ and full body anatomy. Now the scanning methods like your computer tomography, positron emittent tomography, magnetic resonance imaging, all these things have become so efficient, and they are able to do 3D scans with the organ imaging and whole body imaging. These are things going on. I will give you some significance of these studies, how they are going to help in our metabolic perspective. Um, uh, and some again, one more major development which is going on is what is called the Global Human Cell Atlas Initiative. This initiative is started something 10 years back. The idea is to get complete picture of human anatomy at the cellular level, and at the cellular level, that is, we have um, 10 to the power of 13 cells, and these complete each and every cell at the complete location. We people are trying to identify the complete details of metabolomics, proteomics, transcriptomics, everything. This is a very, very big initiative. I don't know how long it will take, but the huge initiative which is going to really expected to transform our understanding of complete human anatomy at the cellular level. Everything, whatever, the whole body omics. You can imagine that. They are going to already there. Some details of some organs are already there. I'm going to tell you that. Then the great thing is the AI doctors, artificial intelligence is becoming very, very interesting. So this is going to compile and integrate the application of clinical knowledge with the metabolism. As I said, the knowledge of the clinician is so huge, and even the knowledge of metabolism is so huge. None of us, no individual nor a group of individuals, can do the integration of this. It's going to be a huge exercise. I think now the computers are becoming very, very smart. So we can use this artificial intelligence to help in developing the integration of this information. And already we are seeing the first level where the AI doctors are coming. That we'll talk about. Then the another major discipline is emerging, which is called the biological artificial intelligence. This is using artificial intelligence, understanding to biological concepts. This is the next level. These are the major things which are going to transform. I'm going to give you some examples of this. Um, First, I will give you how the metabolism now is looking at the anatomical integration context. And a few examples I'm given here. That is the till now what we know is the organs and everything, but we have not bothered about how the organs are coordinated. What is the supply transport takes place? Is that transport is important or not? We never bothered about it. Now we are able to look into that level, and one few examples I'm giving here. Not I'll not discuss all of them in great detail. One is the portal blood, the visceral fat, peritoneum, and epicardium, and cell number. These are the things examples I'm giving how metabolism and anatomy are going to be coordinated together. Uh, So if you look what we know is the liver is one of the major organ, and with respect to carbohydrate metabolism, the insulin and GLP-1 are the major coordinating factors which are important in regulating postprandial glucose metabolism and lipid metabolism. So, it, if you can see in this picture, this is the liver and this is the pancreas. And now we are talking about the intestines here. Uh, so, the whatever food is come here, the glucose, fatty acids, whatever is taken in the diet, these are going to have to reach to the pancreas and liver, and then the pancreas is going to produce insulin, and this insulin will be distributed back to the liver and all the peripheral organs. But now, what is important thing here is that um, whatever is absorbed in the by the intestine, how does it go? You have a separate circulatory system. This is called the portal system. You can see that from all the regions of the intestine, this portal blood is coming, and this is a network is there, and this goes to the liver to pancreas, and there is a connection between pancreas to this. This is a portal blood which is completely different from the normal peripheral blood supply. So the interesting things what has come out is the insulin levels if you measure in the peripheral blood and in the portal blood. When you talk about portal blood, the whatever the nutrients are absorbed are first directly go into the liver. They don't go into the blood circulation. First they go to the liver, all of the metabolites whatever they use, and next they go to the rest of the system. So that is the coordination is there. So the metabolism, that's how the liver is playing the major role. Similarly, the pancreas is going to respond to this and produce various levels of this. So we talk about two important things: insulin and GLP. GLP-1 is produced by at the intestines itself, glucagon-like peptide, and here whatever is produced from here through the portal blood, it goes to the liver and pancreas. And the insulin concentrations, when they measured in the peripheral plasma and the portal blood, you find that the portal blood, the insulin concentrations are very high. That's about the 2 to 4 times higher than the peripheral blood. So that means the insulin secreted from the pancreas first goes to the liver, and in the liver, 70 to 80% of the insulin is degraded. Only remaining insulin, 20% is released to the blood. So this aspect now is very interesting, that unless we are this anatomy is making the distinction for the how the metabolism is. This is important because nowadays, because high level of glucose, you have to give insulin control. In the normal system, it is coming from pancreas. Let's say the 100% insulin is produced, all 100 percent is reaching to the liver, but to the skeletal muscle and the adipose tissue, which are getting from the peripheral thing, they are getting only 20% of the insulin secreted by pancreas. Now, if you give externally insulin to control the hyperglycemic condition, then the you need to give the levels equivalent to what is required for the liver, that is 100%. But at the same time, now in the peripherally you are giving the insulin, so that means that muscle and adipose tissue also get 100%. That's a excess for them what naturally they're supposed to get. So that creates a problem. So that is the perspective, anatomical point of view is giving this, right? Although insulin is acting on liver, skeletal muscle, and adipose tissue, normally whatever is produced by pancreas, only 20% goes to the skeletal muscle and adipose tissue. So that type of thing, but liver requires much higher levels of insulin to regulate the uh, carbohydrate metabolism and lipid thing. Similarly, you have incretins. These are the things which are produced by the intestine. These in whatever they produce here, they are produced by endocrine cells, L and K cells, and this amount, whatever is produced here, incretins have two functions. They immediately act on a vagus nerve, which is present very proximal to the intestine, and then they act on that and they control the food intake. And so, like what I said, pancreas is producing insulin, maximum amount is going there. Similarly, whatever is produced by enteroendocrine cells in the intestine are acting, all major amount is acting on the bigger cells. Now, through this output, whatever incretins, that is, they're going to go to the liver and to the pancreas. And in this, when they're going here, there is a protease present on this endothelial cells called DPP4. In when it is going, these incretins are degraded. So by the time whatever it reaches to liver, it reaches 50%, and it reaches to the pancreas, it is going to be 20%. What we know is that incretins increase the insulin secretion in the pancreas and downregulate lipid metabolism in the liver. That is, but that and they are acting on the vagus nerve. Vagus nerve is having the maximum amount. Half of that is acting on the liver to regulate lipid thing, and out of that again half is reaching to the pancreas, which is stimulating with the insulin. So you can see that anatomy is making a very, very important role in giving the differential role of this thing. This is one nice example. The second example, what I wanted to give you is about the new organ which is present in our body. It's the clinicians know very well about all these components, but as a biochemist, and we are don't deal much with the thing because metabolically they don't contribute a major amount of that. So this also comes as a part of the metabolic dysfunction. Our organs in our body are like in a suitcase. What we pack our all our clothes and whatever items you want to take, because when you're going on a flight or a train or something like that, you're moving around. You don't want to disturb all your components going away. So there's some way of you package them together. So similarly, our body organs are packaged and we keep them in a relatively constant location and everything. That is achieved by a membrane sac. The two types of membrane components are. This is the major one called a visceral sac, and this is what's called a peritoneum. This peritoneum membrane is the almost 1 to 2 meters square area, and you can see that this is all organs are surrounded by that. How it comes up with a very, very interesting development model that some of you are interested, you can do that. So all the organs are bound by this, and this also has a what is the fluid, what is synovial fluid, they put a 50 to 100 ml, and the organs when they're doing the lubricant effect and takes care of that. And this is the anatomy there. And if you look at this, the uh, adipose tissue, we talked about one adipose called subcutaneous adipose tissue. This is called a visceral adipose tissue. And then the we know that visceral adipose tissue is the major one which is involved in the metabolic disorders which is being generated. And it turns out that the anatomical point of view, it looks so small. You know that your intestines is very long, about 15 to 18 feet in size, and all these look there are different regions are there. This is the outer region, the called greater omentum. The intestines are which are connected by this membrane. This is called mesentery, and this mesentery which looks so small. At this, I am going to show you that the regions are the peritoneum sac is about 23 square feet, 52 I told you serous fluid is there, and these mesenteries you have different regions, greater omentum. The greater this larger one is called a greater omentum. This is takes care of many of infections coming not reaching the organs. That's why they call it policemen of the abdomen. Doctors are really they know quite a bit about this. So they call the police men of the abdomen. So if you look at the mesentery, this one connecting with the all organs. What all these the stomach, all the intestinal component, they are being bound together and they are put in this. This is called mesentery. And if you go in the details of that, this is you can see that all these things are connected, and this is the one on this you have the blood supply, neurons, and lymphatic system. All the things are present in this membrane. It is and you have ligaments and everything very tightly holds this thing so that the you can see the regions here. I'm talk about little bit later. These see all these things. If you look at the backside, is they are attached to the near the backside of our body, and then all the intestines are they present in the frontal side. So all these things are coupled by this membrane, and this looks like that. This is the intestines are here. This mesenteric membrane sac of the thing, two things. This is attached to the backside, and you can see that how this membrane is going around the intestines. All the green things are mesenteric, and if you look backside, it looks like this. Okay, so and in the in the very back of your body, that length is about uh, the 15 centimeters, and total length of the thing is around 20 feet, and you can that jejunum it has 8 feet, and ileum it has 12 feet. So this the uh, entire length of the thing is very long. So it is this called is like a Japanese skirt. It starts very start thing here and spreads very long. So you can see the this called root. Root is in the beginning here, and that is very narrow. That is 15 centimeters in length, and this distance is 20 centimeters. But around this, whatever is having total thing, that is about uh, total area which is 1 meter square or something like that. It's a huge coverage is there. And why it is important? And this is what we have seen that when all the nutrients, whatever are going, they goes into this circulation to the blood supply, everything through this. Whatever we talked about, absorption, everything is going through this. So whatever damage is there, these are different organs and things. We know exactly which location, how the supply is going there. And there are, when the lymph is going, there are mesenteric lymph nodes. There are a lot of lymph nodes are present here. And these are the things which are controlling the inflammation, whatever is coming from your intestines. So this is a highly coordinated system. What we now we know the many of the metabolic disorders, when the visceral first thing what we said, most of the metabolic disorders start from this visceral adipose tissue increasing. And now what is emerging is the cells which are present on this, these are called the mesothelial cells. Uh, these mesothelial cells, when there is some sort of a inflammatory things damage what comes in the intestine, the first is getting released incorporated interacting with this mesentery, because you see that large area is there. Mesentery is the constantly getting exposed to the damaging component. So these cells, methicillin cells, they get transdifferentiated into adipose tissue, and they become this visceral adipose tissue. So, uh, and what we also know is that the lymph nodes, they become very large, and the metabolic syndrome, everything goes on, different these things. So now, what's the interesting thing? Because we know the which part of the lymph node is becoming big, that will tell us, and these things happen in a not single time scale. These anatomical changes take place very, very long time. What people have shown is 6 months and 1 year. So the major problem, as I told you before, the biochemical and physiological parameters, these are single time point, they are not cumulative indicators. So anatomical factors are going to be the cumulative thing. And if suppose we know that which lymph node is getting inflamed, that you can see somehow we can see by size or something, then we understand what is going on. And probably we can also now target the thing instead of affecting the entire system, only the location which is getting damaged. So this mesenteric thing is becoming extremely important. Another important aspect of the mesenteric thing is you have lymphatic system. Lymph takes a lot of the chylomicrons, which are all lipid and everything, whatever in the dietary thing, they are being carried out by the chylomicrons, big lipid particles. We know that they go through this, and the damage, whatever happens, is going through. So we have seen two components in the intestines. One is the portal blood. Portal blood, what goes directly is the blood water soluble components, amino acid, glucose, and all those things go through portal blood. Whereas the lipid soluble component, big molecules, they go through the chylomicron. So this we know, we understood the portal blood importance in the previous thing. Now I am trying showing you lipid soluble lymphatic things which are going through chylomicrons. That is going to be so we can understand the lipid metabolism and physiology with respect to this. Okay. Uh, Yeah, this is now these are all pictorial things. What I said. Now what we have is the, as I told you, computer tomography scans are becoming very good. Now you can see the individual thing. The yellow thing is the visceral adipose tissue. Outside thing is the subcutaneous adipose, and you can see the obese individual. He has more amount of visceral adipose tissue, and this is whatever the thing we can show, this is associated with the cumulative effects of physiology metabolic dysfunction. So now what I'm trying to put across to you, because the scanning methods are becoming very powerful, non-invasive thing, we can take the scans routinely. As it becomes more cheaper and more available, then it becomes more possible. So we can whole body scans are available. Analyzing this information at different stages will tell us the therapeutic avenues. Everything will become much better. So that is the which is emerging thing now, and this is the data which has been shown. The in details, whatever the damage, what happens, why the mesenteric lymph nodes get damaged. We can people have demonstrated in terms of exactly lymphatic network how it gets damaged, and the this is associated with the lymph node expansion and so on and so forth. We need not do, these are all invasive things, whatever, but the amount of fat which is produced, and if we can look at this mesenteric lymph node, if we can monitor those sizes, that is going to be give us a enormous uh, input. But knowing the source and place of damage which is chronically taking place, and we can, if we know that, and we know this in stages, how much damage is also taken place, so we can do the treatment in a much better fashion. So that is the second thing. Similar type of thing is there with the heart adipose tissue. I'll not go into the details. The similar way, what we have is that on the heart, you have this called a epicardium and lot of adipose tissue on the heart. Normally, this is supposed to be helping the heart function when it is expanding and doing things. The damage should be minimized. That is taken care of. But if the epicardial adipose tissue this increases more, then it's going to be a problem. These type of things can be now seen by the various techniques which are coming, the which is called the epicardial adipose tissue and cardiovascular disease. Relation is very well known. Uh, now I'm going to show you a similar experiment thing, what people are doing with the CT imaging, trying to show that how the epicardial lipids are getting accumulated, and one can, these things are not exactly do the quantitative measurement, epicardial fat volumes, we can do that, and with that, the you can see the relation between the how much is epicardial fat volume is there, and this is what is the uh, coronary microvascular dysfunction, how much is there, we can understand that. So we can monitor in a non-invasive way very nicely about the cardiac problem. And in India also, cardiovascular problems are now currently in a large scale. It's a very, very uh, serious case. About one third of adults are known to have cardiovascular problems. So the India is really concerned about this metabolic diseases and cardiovascular problems. So now these type of techniques are going to really help in the uh, then these are, I've told you, scanning methods. Now we have what is the cell atlas, huge initiative. This is started in by these two individuals, Avi Regev and Sarah Teichmann in 2016, and some draft of this has come in November 2024, and they are making the data from 66 million cells, and this has been donated by 10,000 donors, and something like 500 projects, 930 labs, 1,700 institutions, and 3,000 scientists are involved in this project. And this is the latest, whatever they have shown. There is a site, international site is available, the information is there. And this, once it is, the information comes, there are a lot of targets are being set, how much to mimic with the human physiology and in terms of clinical pathology and treatment possibilities. Huge thing, this is expected. Okay. Although this is such a huge thing, it's like your human genome sequencing. Human genome sequencing is a just use of human genome sequence have come later. Our last 20 years we are still doing. Now we know that epigenetic population variations in that. So a lot of things have to be inferred after that. So similar thing is, although now this human cell atlas is going to come, maybe in a 10 more years it's going to become a thing, but we should be prepared how to use that uh, into convert into physiological and uh, pathological context. So for that, now this bio artificial intelligence is becoming very important. uh, this is supposed to be an image, generate a generated image of physiology. Uh, you can imagine the computer is now creating the physiology in this. This is the latest uh, picture. And what they are trying to see, how we have to develop a AI for this. There is some sort of a coding problems, which is of a computer domain. Then you have some sort of a inputs of the biological data we need to give. And after this data, we have to ask questions. And then using this interface, the knowledge is going to be, intelligence going to generate whatever information we want to use. People have used, there are many different AI programs are there, and they're trying to use for the biological context and see how much they are going to be validating in various contexts. And you can see that many things are now emerging better and better. Some of the programs are predicting quite well. Some are still not that efficient in there. So these are the going to be programs for specific for different organs. This is where you can see that neuro machine learning program, some sort of a biochemical program. So these are connected with different tissue metabolism formats, and how these different programs are able to succeed. So this is the current state of whatever is emerging, but these are the trends which are going on. So this artificial intelligence is going to take uh, data from the whatever is already present in the proteomic data or transcriptome data, core reactions is going to take. Then you're going to use the basic model of human metabolic model, and then they going to put the context wise. These details we know. They are trying to use all the information which is available in a logistic. This type of thing we need to be done by biologists. The computer programs are there, but how to these models have to be developed? Tissue specific models, enzymes reaction, biochemical model, single cellular model, genomic scale, what level we want to look, signal transduction, whatever is there, these components have to be dealt by biologists. This is our challenge, how to integrate the complete domain with respect to the tissue metabolism and ask the context questions what we want to generate. We are asking the, let's say you generate diabetic model pathways you generate in the liver, or you generate in the skeletal muscle, generate in the adipose tissue. How these pathways, now it may be able to generate much better than whatever our current understanding. So these are the concepts which are emerging. Uh, they're using these AI is becoming much more. All they're trying to use it metabolic input, transcriptomic, genomics variations, all types of things input. They're going to use that, and the efforts are going on. Not only from the fundamental point of view, they're also using the application point of view, how the individuals are responding to drug, what is going on, and so you can the interfaces which is emerging quite well. So uh, ultimately, the idea is they're trying to use this model. If it becomes very good, you can use it as a diagnostic model and also prognostic model. So there are different. These are going the emerging stages of our. Uh, so now the latest issue, I couldn't get the paper there. It's talking about emerging soft medical robots for clinical translation from diagnosis through therapy and rehabilitation. So you can see the how fast the metabolism is changing from the basic concept to the application and overview. There is huge change in this going, and it's going to give you complete. Hopefully, it will help them. So now, with all these inputs, what is the next thing which is going to happen? So this human cell atlas integrating with physiology and disease and clinical knowledge, this is the main application. objective of this thing. Then how do we do that? It has to be integrated with the physiology. Physiology integrate, visualizing physiology in terms of integration of metabolism with time and anatomical context in the health and disease. In terms of tissue, how much time it takes time to grow, like whatever the data we are coming from the scanning models we can show, what time scales are present, and we know the individual cell level lot of pathways we can integrate. That is the way. So in incorporating time concept of HCA in terms of biochemical and cellular process, one unit time scale is circadian rhythm. We know that all components in our body are have a 24-hour cycle. So we have to make a model of each cell, each organ, everything in a 24-hour. Then in terms of with respect to the lifespan of the cell types, metabolism. Some cells are fast dying and other things. So the metabolism has to be integrated. Then, in terms of clinical context, where we know how much time the disease progression takes place, and we know that that is coming from the clinical knowledge. We don't have the biochemist from that. Clinical knowledge, people know the disease progression, and also people know when to do the cure, how much time it takes. That knowledge is also there, but that knowledge has also has to be converted in terms of 24-hour units. This type of thing perspective is there. Probably we are going to have new knowledge of metabolism. So thank you. This is the beginning of the new world of new metabolism from my perspective, and thank you.
थैंक यू वेरी मच सर, बहुत ही अच्छा से पूरा कंपाइल्ड आपने, जिसको रिसर्च करना होगा, जिस डायमेंशन में करना होगा, दे कैन गो इन दैट